Okay, so thanks for joining us today. My name is Georgie Samaha. I am a bioinformatician at the Sydney Informatics Hub. I'm in the bioinformatics team there. We provide the university's research community with support in doing bioinformatics themselves, but also applying these kind of techniques to their own research. We are also an official node of the Australian Biocommons, which, if you haven't heard of it before, it's a national collaborative digital infrastructures project. So our involvement in that, we work with other universities, other research organizations and supercomputing research centers and service providers around the country. And there's a lot of different work going on, but largely, the aim is to make bioinformatics more accessible to life scientists. So I guess kind of, along that vein, today's masterclass is sort of a reflection on my experiences from, when I was a PhD student and I started kind of doing this work and through to the role I'm in now as a professional bioinformatician. The sorts of things that I'm going to be sharing with you today are lessons that I've learned along the way. As I, started out someone who could not code, did not want to code, was thrust onto an HPC without ever having, used the command line before. I reluctantly learned how to code through to, where I am now, where I'm building open source software, doing a lot of project work, consulting with you guys as well. So, I'm someone who came to bioinformatics out of necessity. It wasn't something I ever planned on doing. I guess when I started my PhD, I had a lot of assumptions about what doing this kind of work would involve. In all honesty, I fully intended to process and analyze big mammalian genomes, but I needed to for my projects without ever actually really learning how to code or even use the terminal. Of course, as I've gone through all this kind of work, the assumptions that I've had and my confidence, of course, has changed as I've gotten deeper into it. I've made a lot of mistakes. Some have been way more devastating than others, and I've really developed my skills. So today I'm going to be giving you guys some background and context and really hopefully get you appreciating a bit of the why behind the way things are when you're doing bioinformatics. And then we'll go through some practical tips. So hopefully, you can use these to lean in for yourself, do bioinformatics yourself, apply it to your research, or even just have some context to understand understand the bioinformaticians that you work with, or perhaps even consider getting into this line of work yourself. So we're going to start with some definitions, some information about what bioinformatics actually is, what it involves. We'll talk through a typical experiment, the sorts of data that you're likely to come across, the sorts of things that are involved in processing and analyzing your data. And then we'll move on to some tips. These are basically just, things that I sort of wish someone had shared with me when I was new to all this stuff. We'll have time for questions at the end. So you're welcome to save your questions for then. But if you'd like to, please feel free to use the Zoom chat to ask some questions as well. My colleague Nandan is going to be in there answering questions as needed. So before we start, I just, like to ask everyone to try and let go of any assumptions they may have about, what doing this work actually involves. I personally think the world of bioinformatics is a bit of a contradictory place. It can simultaneously feel both lawless and a bit like cowboy science sometimes. Sometimes it feels like what you're doing is a bit made up. But at the same time, it also seems bound by a bunch of unspoken rules and conventions. And I think that's, largely due to the fact that biology is really complicated. It's very broad. The sorts of technologies that we're applying are advancing very fast. So, trying to rein everyone in and come to some kind of mutual agreement on absolutely everything we do is just not going to happen. So keep in mind that as I talk through this stuff today, keep in mind that, we are all applying bioinformatics to very different scenarios. So we're working with different organisms, different research questions, different technologies. And unfortunately, that means there are going to be some pretty clear inconsistencies in how we design our experiments, the quality and the type of reference material that we use, sorts of reference genomes and that sort of thing that we use. the sorts of computational resources and platforms that we'll also be using, and the sorts of community standards that we'll be following as well. So I guess in keeping with that, I actually found it kind of hard to come up with a neat definition of what bioinformatics is. I guess at a high level, it is the application of computational tools to processing and analysing and interpreting biological data. More specifically here, we are pretty much only talking about data in the context of molecular biology. So, think nucleotides, proteins, amino acids, metabolites. It's an interdisciplinary field that involves computer science and biology, as well as mathematics. You definitely, don't need to be an expert in all these things. I definitely think you need to have a strong biology background, but you don't need to be an expert in all these things in order to apply this stuff to your research. So, bioinformatic data sets, I'm sure you all know, have become essential to the molecular life sciences over the course of the last 20 years or so. It's obviously bioinformatics has been around for a lot longer than that, but in the last 20 or so years, since next generation sequencing really popped off, things have become a lot more accessible and comprehensive. So high throughput techniques, so I'm talking about, next generation sequencing and things like mass spectrometry. Those techniques have really significantly expanded our capacity for data generation, and they've really provided us with a lot of information on the molecular constituents of biological systems. And along with that, we've also learned, there's been a lot of advancements in computational power and the sorts of algorithms that we apply to this work. So it means that we're able to do far more sophisticated analysis and model way more intricate biological phenomena as well now. So depending on, your background or, what kind of research you do, you probably have come across certain buzzwords related to bioinformatics. These days, I feel like most of us have definitely heard terms like SNP or personalized medicine, or, even done a DNA test or something like that. In reality, bioinformatics, in my opinion, it's not all that sexy. There are a number of really dry and complicated, but essential and I guess unavoidable aspects to the work. Firstly, there are the struggles and responsibilities of working with big and complex data. So, there are a lot of research disciplines that work with, quote unquote, big data. So things like radio astronomy or climate simulations, that sort of thing. But I guess unlike those disciplines, bioinformatics data is not just big, but it is also highly heterogeneous and very noisy. So, that adds a real layer of complexity to the kind of work we're going to be doing. Next, there are, of course, the data privacy and governance restrictions that, may or may not apply to you. Of course, it really depends on the source and the type of your data. Navigating all the rules around compliance and the ethical guidelines that you might be up against, that is a significant and often overlooked, I think, aspect of bioinformatics research, especially when you're dealing with highly protected data. So you're dealing with, things like human genomes, that sort of thing. Finally, there are also the struggles of code management. So, a lot of us don't have formal computational training when we start out doing this work. So ensuring your analyses are reproducible, of course, is really essential for the credibility and reliability of your results. And so, I guess that the need to manage our code properly, but the fact that we aren't properly trained in doing this stuff often means we come up against challenges with documenting and maintaining and organizing our code as well. And these things are, of course, crucial for replicating your results and collaborating with others as well. And on top of that, you're usually juggling multiple tools or packages in order to run just, a few small steps. And handling things like installation of all those things can be quite a massive burden. So if we start off by thinking about what a typical experiment looks like, again, of course, remembering we're all applying very different data types, different analyses and processes. We're all largely following the same research protocol. We, of course, start with study design, where we decide what kind of data we need to answer our research questions, the specific processes that we need to apply to the data to extract some nice answers for our research aims. The sorts of things that we really need to think about at this stage is things like the number, type, and quality of samples. Do these samples reflect the biological context of your research questions? Do you have enough samples for statistical significance? And also things like, does the type of data that you plan to collect, whether it be things like, whole genome sequence, some RNA, some protein, maybe you're using a genotyping array or a microarray, does that data set actually have the capacity to answer your question? The next step involves digitizing your chosen biological material. So, whether or not you have extracted DNA, RNA, protein, or metabolites from some samples, you need to convert that material into a format that can be analyzed by a computer. So, during this phase, what comes out of a sequencing machine or a mass spectrometer is typically pretty fragmented and noisy, and this necessitates the next step, which is the data processing step. So this is where the bioinformatics really begins. During this phase, we convert that raw sequence data that maybe comes off the sequencing machine into a format that can be analyzed. So, if you have sequenced some DNA, that goes into the sequencing machine, comes out in some short little sequences. Those sequences then need to be, say, aligned to a reference genome or something like that, or assembled into a contiguous sequence, so that it's in a form that can adequately represent the biological sequence that we're then going to analyze. And this encompasses all sorts of things that we're going to talk about in a bit, but really the focus of our analyses here is to identify patterns that can provide us with some insights into the biological questions at hand. And then, of course, once we have those potential insights, we can employ various techniques. Some of them are computational, some of them are a bit more manual. But yeah, we can employ various techniques to contextualize our results and ensure that they actually reflect the biology that we're looking at, and can also help validate our results as well. So this slide may seem a bit obvious, but it can actually be a lot harder than you think to get right. We get questions from researchers all the time about, what data is right for this question. And sometimes even, unfortunately, we get people coming to us after they have collected and analyzed their data, and they ask us why they aren't getting the results they expected. There are, of course, lots of different types of data that we can use to give us insights. But it's really important here to keep in mind that your choice is, like I said before, it's relevant and it reflects the biological question or your hypothesis that you're trying to address. So say, for example, you're looking at some gene expression related to a heritable neurodegenerative condition. A couple of things that are really important for you here is, you're not going to be able to extract meaningful information about the condition from RNA that is taken from a non-neuro tissue type. So say you take RNA from some skin samples or some blood, you're probably not going to be able to make adequate assumptions and get some nice clean results about neurological function from those tissues. Nor could you expect to, yield statistically significant results if you were to not include things like biological replicates or not enough biological replicates in your sample collection stage. On top of that, you're also going to need to ensure that you're collecting data that is high quality. Poor quality data can always introduce noise and therefore reduce the statistical power of your work. So it's really important when, say you're extracting that RNA from a tissue, that you try and avoid things like DNA contamination as much as possible, and you try and meet the minimum yield of RNA, recommended by your sequencing provider or something like that. I mean, that being said, sequencing data is very high quality these days, and a lot of the tools that we use to process our data are able to handle poor quality data quite well. Another thing that you need to consider is the cost efficiency and working within your budget, of course. Sometimes, it might appear to be fiscally prudent to reduce the number of samples you collect, but in doing that, you might risk reducing the statistical power of your analysis. And, This all can be really hard stuff to work out for yourself sometimes, so you can always come and talk to us. We have a statistical consulting team as well that are really very helpful with helping you work out things like power analysis and working out, collecting the right number of samples for your projects as well. The final consideration here are the ethical considerations. You've just got to remember that it is your responsibility to adhere to the relevant ethics agreements and guidelines. It's especially important for those of us who are working with that highly protected data. There are lots of rules around how you collect, store, and manage your research data storage. So just make sure you're across that as well. So of all the data types that are commonly used in bioinformatics, you'll come across and you'll hear a lot of ohms and omics terms. These refer to high throughput technologies. So these are things that come from next generation sequencing and mass spectrometry, and they capture the complete set of a particular molecule and maybe a tissue or an organism. There are a lot of ohms out there, of course. I've just simplified things here a little bit, and I'm just talking about the ohms that correspond to those basic molecules, thinking back to the central dogma, right? So remember, DNA is transcribed to RNA, RNA is translated into proteins, and we can think about things like metabolites maybe as like a product of and regulators of this process. So if we compare these like omics technologies and data sets with more selective approaches, like targeted approaches, like microarrays or genotyping arrays, these techniques are going to be more holistic. They might be really useful where the aim of your research is not just to focus on particular genes and proteins or metabolites, but also maybe on more complex aspects of regulation, of structure, structural organization and interactions as well. And also if you're trying to perform kind of novel discoveries, arrays tend not to, they contain like a very narrow set of data. So if we consider a research project just as an example where people are maybe studying coat color genetics in cats, we can think about the unique perspective that each of these data types provides. So if we were to sequence, say, the genomes of a number of cats with different coat colors, we'd be able to discover specific genetic variants, maybe novel genetic variants that are associated with these different coat colors. And depending on the way we collected our samples, maybe we can infer some information about mode of inheritance. However, we're probably not going to be able to infer much functional information from this. For that, maybe we're better off looking at something like gene expression levels from transcriptome data or some protein abundances from the proteome. But for those approaches, they have to be tissue specific, remember? So in this case, we would be collecting data and sequencing data specifically from something like the hair follicles. And in doing that, we might be able to identify how differences in the genes, in certain genes, affect the expression levels and the proteins that they produce. So from transcriptome and proteome data, we can infer some of those regulatory and functional elements that might be affecting key structural proteins and enzymes that might play a role in something like melanin formation. Beyond that, things like analyzing the metabolome could help you clarify things like pigment synthesis pathways or metabolic activity and how this varies between the different groups. I'm definitely not saying here that you need to collect all of these types of data sets, to have such a comprehensive types of analyses, but it's just to highlight their unique applications. And that being said, there are, of course, multi-omic techniques are increasingly popular, and they've been, become a great way to kind of address different layers in your research question as well. So once you've decided on the type of data that you need, you're going to need to find it. You don't just need to find the data that you're going to use to, the data that you're going to be analyzing. You're also likely going to need a bunch of complementary and supporting data sets to help you process and contextualize and interpret your primary data set as well. So if we first focus on, how you can collect and where you can find primary data from. So you can get this from a few different places. You could go to a sequencing company or a mass spectrometry facility and pay them to generate some data for you. So, sequence that biological material for you. Alternatively, or perhaps to, I guess, complement the sequence data that you've generated and increase your sample numbers or something like that, you can also find others published data sets for analyses. You can find raw data online. You may have noticed, that increasingly a requirement of publication is that you release your data publicly. And when people do this, they tend to release their data on platforms like Sequence Read Archive, European Nucleotide Archive, and the Gene Expression Omnibus as well. So there's a lot of different places for you to find raw sequence data. It's not, you don't have to just generate your own all the time. If we move on to the next kind of category, so that's supporting data. So to support your ability to analyze and contextualize your data, you're also going to need things like reference materials. So that includes things like reference genomes, gene and variant annotations as well. These sort of things are publicly available, but do keep in mind that unfortunately the availability of these sorts of data sets and references are often kind of limited if you're working with, say, a non-model species. There are plenty of workarounds for this. You can, there are people employ things like cross-species applications all the time. All in all, though, there is a wealth of public resources available to you in analysing and contextualising your research. So all of those different, raw sequence, reference, and annotation data sets, they come in a variety of formats. These formats generally adhere to a few different standards that have been created and are maintained, and they are updated sometimes by the bioinformatics community. So having these standards ensures that the kind of data that we're using is consistent and it's also interoperable. So that means that it can be used as inputs for multiple different tools, and you can trust, the formatting that you're going to get with those files. The definitions of some of the more widely used standards, so you've probably heard of FASTQ, VCF, BAM files, or SAM files. These are public standards. You are able to very easily Google and find out exactly what those standards encompass and what those files look like. But if you're new to this stuff, it can definitely be challenging to get your head around all these formats. They're actually not as complicated as they seem. I know that sometimes they look quite terrifying, but generally these files are just text files. They might be compressed, but they are just text files. So that means they are human readable. Often they consist of maybe a header section with or sections that contain some metadata and describe some characteristics about the data. And then they also contain, the actual biological data themselves. So if you're thinking of something like FASTQ, for example, for FASTQ files, each read is going to have a little header line that gives you some metadata and quality information about and sourcing information about whether about that read. It's going to give you the read itself, and you're going to get some quality scores for each base in that read as well. It is important that you get familiar with these formats if you're going to use them. And get a feel for what they contain and where the pertinent information is in them as well. So if we get back to that kind of experiment workflow, now that we've kind of handled the important attributes of data acquisition and we move on to data processing, I guess the same thing as I said before, even though we might be dealing with very different data types and formats, the general steps that we're going to need to take to prepare them for analysis are pretty consistent, especially when we're starting from say, raw sequence data. Generally, we need to assess the quality of our data and identify any issues that might compromise our analysis. We then kind of clean it up, filter out any low quality data points, remove some noise that might interfere with our ability to yield accurate results. And remembering for this part that, our sequence data usually comes in that fragmented, chopped up form. We need to reconstruct it to, infer that original sequence or structure before we can then identify some biologically relevant features. So on the right here, I've just got two examples of what this might look like for genomic data and proteomic data. While, things that are happening under the hood are actually very different in each step, you can see that we're still following the same workflow. And, a practical consideration here that you might not be aware of if you haven't kind of done this work before is that we tend to treat these steps as separate from one another, and we generally need to use generally need to treat these steps as like individual blocks. So we use different reference or supporting data for each of these steps, and we tend to use different pieces of software to complete each of these steps as well, because, in themselves, they are quite complicated and involved. So once we have our analysis ready data, we can get on with applying some statistical methods and algorithms to infer some insights from it, right? So this can obviously cover a huge array of different methods. No matter what method you're applying to which kind of data, a few essential considerations for you are to ensure, firstly, that the methods accurately reflect the biological reality and that they are statistically robust. Another thing to consider, and this also applies to the data processing step, is the impact data complexity and scale and size can have. So given how big and complex these data sets are, and how complex the algorithms we apply to them are, these data sets and their analyses can be really computationally greedy. So by that, I mean they need large amounts of resources, so probably large amounts of disk space to store the data. So, your raw data might be quite large, but you'll also be generating a bunch of intermediate files that themselves can also be large. And you'll probably also need things like a lot of RAM and probably CPUs as well to process your data in a timely fashion, I guess. So, this is something you definitely need to consider more when you're working with large-scale data sets, whether that be things like organisms with really big genomes or really large sample sizes as well. So generally at the end of this, so we've processed and then analysed our data. Generally at the end of this, we're going to have a set of features that we, are particularly interested in. That might be, a list of genes that house, variants of interest, some proteins, things like that. So we need to be able to link these features to the relevant biological processes, cellular components, and molecular functions. So, we can interpret and make assumptions about or hypotheses about, the sorts of impact that they are having. I personally find this step to be the most fun. I think it's, very much like detective work, but it is definitely really hard work. It's, a lot of problem solving. It can be quite manual and not very straightforward. And it often involves a lot of searching online to, kind of tie all these things together. But, there are certain resources available online that help us connect our results to all sorts of functional annotations. So that includes things like gene ontologies. So gene ontologies are kind of functional and structural categories that we can use to group genes into general themes. It also includes things like metabolic pathways that we can use to, enrich links to biological processes. We can also look at things like where our results sit in the broader population. And this is especially useful, when you have some mutations or some variants of interest. And you want to know whether or not the thing you're looking at is, common or rare in the population or pathogenic. So, if something's common, it's probably unlikely to be pathogenic or, deleterious or something like that. And, in terms of determining if something is damaging as well, there are a lot of resources out there for things like pathogenicity scores and that kind of thing as well. Again, here, just like with the reference and annotation data sets, these sort of functional annotations are they're not widely available for all organisms. Humans are typically best supported, and, mouse and then bacteria and things like that. So things that are more clinically relevant are probably going to be best supported. But, there are plenty of workarounds. You can definitely get creative with this type of thing. And you can infer a lot of stuff using annotations that might be available for closely related and maybe better supported species to the, the data that you're working with. So now that we've covered all of that, we have the inevitable question of where you can do this work. So, of course, there are a lot of different interfaces and infrastructures for us to work on, and you're often, going to need to use different infrastructures and different interfaces, interchangeably within the same. Sorry, not interchangeably, for different purposes within the same project. So generally, we can group these two things into graphical user interfaces and command line interfaces. And there are some, some things to consider when you're, things to consider when you are choosing which one to use. So graphical user interfaces are also called GUIs sometimes. They can be open source or freely available, but they can also be commercial offerings. They're really great for, beginners or those who are maybe less familiar with coding. They do often, some of them do offer like no-code solutions for this kind of stuff. They're really great for offering like integrated visualizations and definitely a more user-friendly approach. However, of course, something to consider here with these features, with these nice accessible features, definitely comes some trade-offs. So they tend to be suited better to smaller scale work that is less computationally intensive. Typically, you can install a lot of these tools on your own computer. So, we're talking about things like RStudio or maybe CLC Genomics or something like that. So installing them on your own computer, for the most part, means that you are using the compute resources that are available on your local machine. So your RAM, CPUs, and disk space are kind of limited by what's available on your local machine. If you're working with, say, web-based GUIs like Galaxy, which a lot of you may have used before, you will have access to some external computing resources on the back end. My understanding is that Galaxy is a whole lot of national HPC. So you'll definitely get access to more compute resources. So you might be able to do more computationally intensive work, but you are still kind of limited to the sorts of work you can do. In contrast, command line can be a really intimidating place to work for novices. It's definitely got a steep learning curve. While you can also use the command line and a terminal application to, use the resources on your own local computer, you will definitely have to be using a terminal and the command line to access infrastructures like the HPC or a lot of cloud computing platforms as well. Using the CLI, you're definitely going to need to know how to navigate the Unix environment. So a bit of basic bash is really helpful. You're going to have to do things like manage your own environment and manage your own software installations, and quite a few things that can be quite painful. But all of that can really pay off in terms of the amount of flexibility and versatility that you'll be getting here. You'll definitely be able to customize your work a lot more than on a GUI, and you'll definitely be able to perform much larger scale work. So I know that's probably a lot of information to take in, and I guess something I found challenging when I started this work was that I felt like there were a lot of unspoken rules about how these things were actually done, how each of these steps, how I could do that kind of work practically. And of course, as biologists and clinicians, we don't receive formal computing training. So to help you implement these things, I just thought I'd give you guys some practical tips to wrap up with. And, these are things that I think have definitely made me feel more comfortable about doing this kind of work. So my first tip here is that there is no one-size-fits-all solution. We talked a bit about this at the beginning, but, I think it's really important to remember that the diversity in our data and applications means that we're each facing quite unique circumstances, which may require a slightly customized approach. This is something I definitely didn't appreciate when I started out during my PhD. I, naively looked at things like tutorials online that, described things like genome alignment and variant calling. And, I honestly, didn't give much thought to the runtime and that kind of thing, how much work it would take, how much time it would take for me to run these jobs. If I'm working with, say, a big mammalian genome, like I was working with some big cat genomes, I didn't really give much thought to the differences between running a huge genome versus running something tiny like a bacteria or something like that. It sounds silly, but that's where I was at. So yeah, I think it's really important you guys consider the range of genome sizes and complexities across different organisms. Things can get very complicated even when you're working with this in the same class. There's a huge, even if you think about mammals, there's a huge range of sizes and complexities within mammals. This is going to have a dramatic effect on the compute resources that you need to process and analyze your data. These differences are definitely going to determine where you can work. So whether you can work locally on your laptop, or if you'll need to access something like an HPC or some cloud computing. So some scalable resources, really big resources there. On top of that, your experience level or your coding ability might limit where you feel comfortable working, and it might mean that your processing is quite a lot slower than, say, someone who has access to big computers, is comfortable working on an HPC to run their stuff and working in more flexible environments. My next tip is to not feel like you have to reinvent the wheel. I think this work can definitely feel a bit lonely at times. It's especially lonely when you're the only person maybe in your group doing bioinformatics. Despite how that actually feels, I promise you are definitely not alone. The bioinformatics community is actually very collaborative and open. I really think it's just a matter of knowing where to find help. So there are lots of communities and resources out there for you. If you are troubleshooting an issue or some broken code, trying to plan an experiment or learn a new technique, I pretty much guarantee you that you will be able to find the answer to your question or to your problem online somewhere. If you have questions or you need support, you can always come and talk with services like us. But you might also want to engage in some less formal chat. You can engage in online forums like BioStars or Stack Overflow or Seek Answers. I can't tell you, I think pretty much every time I'm Googling my way through a problem, results will come up. So conversations, discussion boards from these forums will come up in my Google results, guaranteed every time. There's also a lot of training material and free events online that offer really valuable opportunities to build your skills. As I mentioned at the beginning, we are a part of the Australian Biocommons, and we're a part of their training co-op. And they offer online events that are free and open to all Australian researchers. And there are also plenty of other places to find training events and resources as well. So there's the Galaxy Training Network, the European Bioinformatics Institute, that EBI, they also provide great self-directed training materials on a diverse range of different topics. In terms of actually doing your data processing and analysis, you definitely don't always need to be writing custom code. There's a lot of great software out there, and the bioinformatics community really prioritizes open source software. So it's all freely available to you. And yeah, we touched on this before, but managing software is one of the biggest struggles that you're going to face when you're doing this work. In my opinion, I always struggle with it. So I highly recommend, where you can, trying to use software management tools like containers, R packages, and things like Bioconda as well, to help you manage installation and dependency clashes and things like that as well. You also definitely don't have to write your own workflows. So if you're doing, say, a standard data processing workflow that uses, you just want to use the community standard best practice tools. More and more these days, people are sharing their workflows and using workflow management systems like Nextflow and Galaxy to write and share their workflows as well. And even for things like writing your own, custom code or workflows, you can always reuse and borrow other people's code on platforms like GitHub and Bitbucket. Exploring and using other people's code can really save you a lot of time. It can offer you a new perspective on how to approach a problem, and it can definitely prevent you from, wasting your time and reinventing the wheel. So, if you're going to engage with some of those support communities and the services that I just mentioned, something I think is really important here is learning how to ask questions properly. So, keeping in mind that there are lots of different ways to do the same task. So you will need to be specific about, your circumstances when you're asking for help. So, I've just got 3 situations here. So say you are, troubleshooting an issue. Maybe you found a bug in someone's tool, or their documentation isn't clear about how to run it, and you're running into this bug that you just can't get over. Your best bet is to contact the person who wrote that tool or that code. A lot of bioinformatics tools, their code bases are available, and they're all very visible on a platform called GitHub. People frequently, use the issues feature in GitHub. It's, very transparent. They log issues there. You can see them. There's a big log. The history is all available. You can submit issues there if you'd like to. And it's a good way of direct communication with a developer. The best way for you to get an answer to your question from a developer or someone who's trying to help you troubleshoot your code is to provide specific technical details that they will need to reproduce your problem, understand the source of your issue, and help you find a solution. So really important to think about things like, tell them the version of the tool you're using, show them the command you use to run it, show them the error logs. That kind of thing is really helpful. If you have some general questions about a method or your experiment, or you might want to get involved with some of those community forums like BioStars. Here again, I think context is key. Asking specific questions, listing out the important considerations is the most likely way for you to get a discussion going and get the answer that you're looking for. If you're just looking for some general support, you can also, of course, always go and talk with a bioinformatician like me and my colleagues at SIH, or maybe someone else you work with. But again, the same stands as the previous, it stands here for the same as the previous 2 points, will be much more useful to you if you can provide some context, and if we can understand what you're trying to achieve and what your circumstances are and the problems you're facing. Just remember that, everyone does things just a little bit differently. There's a lot of variety in what we're doing. So continuing on from that, if you are someone who is considering to learn to write code, keep this next tip in mind. Sure, if you become a bioinformatician who develops tools or workflows, you're going to need to learn to write clean and efficient code. But as a researcher, if you're just starting out or you're just dabbling, your main focus should really be on ensuring that you can understand and reproduce your methods. I personally think of coding as a means to an end. I, am not someone who's particularly enamored with, the intricacies of Python, like some of my colleagues are, or, any other language that we're using. I started, learning how to do this kind of stuff. I started with some basic bash that helped me navigate the command line. I then learned bits of R and Python as I needed to apply them, to practical situations like extracting data from files or creating visuals. So starting small and, with small manageable tasks is a great way to start out and build your confidence before you tackle more complex challenges like, say building a big workflow or something like that. Another thing that I think is particularly important in the researcher context here is to prioritize functionality in your code. So don't worry too much about efficiency until you really need to. As you gain experience, you can definitely refine your code and learn how to optimize things. But, your focus really initially should be on developing and understanding the logic of your code. So just getting it working at a basic level. And my final tip is about managing your methods and your code. This is something that I'm sure holds true for any lab work you're doing. It's to not trust your future self to remember anything. So, you should be documenting things as comprehensively as you can. I treat this like I would treat lab work. There are a few ways that, you can do this. So you can write comments throughout your code, so you know what each line is doing. You can also keep a notebook that, documents your choices and your issues and justifications. There have been so many times where I have spent hours troubleshooting something. I tell myself I'm definitely going to remember what happened, remember how I resolved it, because it was so annoying. And, within a few weeks, I've forgotten everything. I'm somehow gotten myself back at the original starting point of facing the issue, and I'm going through the same process again. So, you just waste a lot of time if you don't document things. In addition to documenting things, you can, of course, manage issues within your code by doing things like implementing version control. So ensuring that, you're using the same tool version to run a script. You wouldn't maybe necessarily think it, but the commands you can run vary between tool versions sometimes. I don't really understand it. You might find it's particularly an issue with something like GATK. Another thing you can do to kind of manage and version control your code is to, save it to platforms like GitHub, so you don't lose it or write over it. And of course, you can, implement things like tests into your code as well. So, there's just some basic, pretty simple things that you can use to save yourself some time in the long run. So that's, the end of the presentation. The intention here was really just, trying to get you guys thinking a bit more broadly about this work. I think my biggest takeaways for you are to remember that bioinformatics data it's big, it's complex, and it's messy. And that means that, we often need things like big computing resources, complex algorithms, and a lot of different tools to get things working. And so, just to remember that, a lot of mess comes along with that, and it's not always very pretty. The resources, the tools, and the data that you use are going to be totally dependent on your experiment and your research questions. It's really important that you're careful about collecting the right type and amount of data to ensure that your results are relevant to the biological questions at hand. And remember, you're generally always going to need to clean, process, analyze, and contextualize your data. So, no matter what your application, and that there are lots of tools and resources available online to help you do that. It just might not necessarily be obvious or clear what those tools and resources are and where they are. So, if you feel a bit lost, talk to people who have some experience, Google things, and, have a go. So thank you very much for coming today. If you'd like to, you can always get in contact with us for support with your research. I've got our bioinformatics team on the screen here. We're always happy to have a chat and help you out, hear about what you guys are working on, what kind of issues you're facing. We do offer things like consultations, fee-for-service project work. We've recently started rolling out a core workflow service. So, if you need to, we can process whole genome sequence and RNA-seq data for you. You can get in contact in lots of different ways. I, highly recommend if, you're looking for some advice ever, you are always welcome to join in and just to our monthly happy hour session on Zoom, just for a chat or some advice. So thank you very much. I think we've got,